Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started.
Okay, welcome everybody. This is our very first ever live podcast for the Genetics Podcast. I think some of you may be longtime listeners, but this may be brand new to others in the audience, so thank you for joining us. I'm following the number one rule of podcasting today, which is if you have exceptional guests, then the rest takes care of itself. So I'm really excited to be joined today by Heidi Rehm, who's Chief Genomics Officer at Mass General Hospital and Co-Director of the Medical Genetics and Population Genetics Program at the Broad Institute. And I'm also excited to be joined by Slavé Petrovski, who's Vice President of the Center for Genomic Research at AstraZeneca. We're going to hear from them about what's most exciting to them right now, some of their recent impactful work, and also their perspectives on the exciting opportunities in the future of the field. We're going to have time at the end, 10 or 15 minutes for the audience to ask questions. So if you're thinking of interesting questions along the way, then feel free to be brave, stand up, and you'll end up on our podcast feed as long as it's a friendly question and not just a comment, which you're not allowed to make at these kinds of events. So without further ado, I'm going to ask Slavé first and then Heidi to just give a little bit of background of themselves, the kind of work they're focused on for people who aren't super familiar with them yet. So Slavé, over to you.
Thank you, Patrick. And it's an absolute pleasure to be here. I was talking to my team and saying, I think we hit the big time. We're on Patrick's podcast. No, jokes aside, so I joined AstraZeneca back in 2018 now. And at the time, AstraZeneca announced a company-wide genomics initiative, which was motivated by the belief that, what better model to define our therapeutic strategies on than human biology. So we set out to build a comprehensive human genomics and multi-omics resource within AstraZeneca that allows us to answer various important questions across the R&D value chain. And today that team's a global team that's really looking to see how we can leverage human biology from genomics to transcriptomics, proteomics, link clinical data, imaging, all those modalities to help us discover new targets, better understand patient stratification for clinical trials, identify the right diseases and within them the right patients for our medicines, and even to be able to find the patients who benefit from our medicines much earlier on in the disease process than we currently do today using traditional approaches. So that's a bit of a summary of the kind of ways that we're using, and I'm sure we'll get into deeper dives throughout.
Heidi?
Excellent. So Heidi Rehm, I work both at the Broad Institute, where I'm both, as Patrick said, co-direct the Medical and Population Genetics Program, but also one of the major hats I wear is as a clinical lab director, and I've done that for over 2 decades. Now as the clinical lab director at Broad Clinical Labs, where we offer clinical sequencing and interpretation, things like that. So I partly wear a hat as a clinical genomicist, and then I spend a huge amount of my time in rare disease research, gene discovery, and that area, and then also building resources for the community, whether it be the ClinGen program, the Nomad project, Global Alliance, Matchmaker Exchange, all these sort of platforms that we build to support the community in the clinical genomics space.
Amazing. I'd like to start with both of your perspectives on what are the most exciting things for you right now in either the research realm or clinical application. Let's start with genomics and we'll turn later to proteomics and some of these other emerging technologies, but I'd love to get your perspectives there. Maybe we'll start with you, Heidi.
So I would say excitement is on 2 dimensions. One, literally an hour ago, we announced that we'd won the Guinness World Record for the fastest sequencing, diagnosing a babies in the NICU in 6 hours for a full sample to clinical report, with the sequencing being 3 hours and 57 minutes. So like bringing the technology to a place where we can really use it in immediate clinical questions, I think is really exciting. But this may, and those kinds of things are obvious, advances in technology, they sort of drive our field. But the truth is what really, makes me excited is, I've spent the last 2 decades trying to move our field forward through much more laborious processes of getting people to share their data, because so much of what we do, we can't interpret, variants for patients with rare disease. A third of every patient tested for diagnostic purposes end in an inconclusive result due to a variant of uncertain significance. It plagues the patients, the physicians, the researchers, everyone. And 2 decades ago, I was begging labs to consider sharing their data openly, and working on ClinVar with NCBI and getting people to submit, and it was hard. And now, I particularly wearing my hat as the chair of the Global Alliance for Genomics and Health, I have people just regularly coming to me and be like, how can I share my data? Help me do it. And launching new platforms where everybody wants to actually share their data and they understand the need for it, the benefit of it. And to me, that is revolutionary, beyond any technology. Obviously, technology enables us to do those things, but the mere fact that the culture has changed demonstrably is something that is so exciting to me. Slavé.
Well, first off, congratulations on the record. Gosh. So I think very similar lines. If you think about, and I'm glad Heidi's here actually, 10 years ago, GNOMAD was transformed medical research for those purposes, understanding variants that are general background population variants versus ones that might be clinically relevant. But even GNOMAD itself back then, I think, was about 60,000 samples and not linked to clinical data. It was just kind of a variant frequency. I say just very, very politely, because I think it had profound impact in terms of clinical diagnostics. But we live in a very different world today in terms of data access, the scale of data, the diversity of data that's available from humans. So we're no longer dependent on simple models, cellular models, etc. We can actually learn about human biology from humans at a scale that's wasn't even imaginable 5 years ago. We're talking about millions of human genomes with hundreds of thousands approaching in terms of other complex modalities of dynamic omics like proteomics, etc. And we're now at the cusp where the technology is also getting to a point where it's becoming a really important enabling tool. But we all talk about AI, I think, and I'm sure this will come up throughout the podcast, because I think everybody talks about AI. It's a very timely discussion. For me, though, I think that the key catalyst for a lot of the transformations that we're seeing is perhaps more the data that's available today that wasn't available 5 years ago, more so than necessarily the new algorithms, the new technologies. They definitely help and they enable the new discoveries, but without these data that we've been generating in the last decades, we wouldn't be able to make a lot of those breakthroughs.
Yeah, I want to get both of your perspectives on that, because I think we selected you two for a reason, because you represent both a research angle in both cases, but also clinical and large pharma apparatus investing heavily in genomics. Heidi, I'll start with you. Were you the first chief genomics officer, or certainly one of the first? How do you think about infusing genomics through a healthcare system, and also making sure that the data you generate there can be shared and used in research? And then Slavé, similarly, thinking about clinical trials and biobank partnerships, where you all have been driving the scale to probably approaching 2 million samples in both of your cases, right, if not more. So how do you think about changing this from a small cohort or biobank-based study to infusing it into our day-to-day work and making it just part of the fabric of how we collect data?
Yeah, so I took on that Chief Genomics Officer role, I think it's now 8 years ago, and I left, I was the clinical lab director for Mass General Brigham Laboratory for Molecular Medicine that I built and was at for 17 years. But the truth of it was, I realized that at the lab, we could offer every genomic test, whole genome sequencing. We could diagnose, patients with the latest technologies, but so many patients were not getting access to these tests. And so I essentially switched and started working on the front lines with all of the, I formed the genomic medicine unit and a genomic medicine implementation team where I worked with, at the time, Katrina Armstrong, who was chair of medicine at Mass General Hospital. And she helped me find, a lead for every clinic and every division and department across Mass General Hospital, bring them into a team and say, how can we ensure that every patient in your clinical specialty can get access to genetics and genomic testing as needed? And it was amazing to me to be at one of the world's most amazing hospitals who, some departments weren't using genetics at all, despite the fact that they were seeing patients with genetics disorders. And so just helping them figure out how to incorporate genetic services into care was a huge need. And we worked very closely. Subsequently, we've now launched a strategic initiative with primary care, because I find that not I, but we as a whole society have insufficient referrals to specialty clinics for underrepresented populations. They don't get there. And so you also need to work with primary care and help them embrace genomics. And so some of the most common clinical testing that should be done and can be done, like cancer risk screening, if you meet NCCN guidelines for cancer risk, or carrier screening for reproductive risk, should be just ordered at primary care, right? And don't have to get them to specialty clinics. So with the help of a primary care physician who was very interested in genetics, Leland Hall, we launched, a practice of working with all the specialty or all the primary care clinics to offer genetic counseling virtual services to help consent the patient and get the sample. And we would just pair the primary care physician. They'd still order it, but we'd help them with the pieces they weren't familiar with. And those kinds of things, I think, are really critical in actually allowing genomics to be part of everyday medicine, which is still a challenge with our insurance barriers, but I think we're making some headway.
Yeah, and so remind me the question from the industry perspective.
Yeah, how do you move, or how have you moved from being a consumer of you've driven sequencing in the UK Biobank along with a consortium of partners, but also sequencing through clinical trials, through the whole apparatus that you have as AstraZeneca, and what can people in the audience who may be in biotech or pharma learn about how to make it part of the drug development process from beginning to end, in the same way that Heidi's thinking about how to infuse it through healthcare?
Yeah, so when we set out this journey, we thought about what are the key sources for which we can get access to research participants for advancing our understanding of human biology. And one of them was our clinical trials, and from 2017, all of our clinical trial participants get offered an option to be part of this broad research consent form, which allows us to study the outcomes and interventional components, which are very precious and not very accessible in biobanks and other settings. We complement that with partnerships with leading academics across the globe who've invested sometimes decades of their careers, building up precious cohorts with longitudinal, multi-specimen sampling that provides deeper dimension into specific disease biology. And then, of course, the more biobank setting, which is increasingly being considered quite precious in terms of understanding the genetic prevalence of diseases at the population level, more so than just at the ascertained clinical. And I think one of the reflections that I'd have is, the dependency that we have on emergency services and hospital admissions for diagnoses is creating a huge problem for the healthcare systems. To Heidi's point, if we can get a lot of these diagnoses happening earlier, the other advantage of that is that people can start getting the necessary care earlier. And I think we all recognize that for many of these diseases, the earlier the intervention, that's not necessarily always therapeutic. There's lifestyle, there's other aspects as well of that. But the earlier the intervention, the more likely the probability that the individual will live a longer, healthier life. And of course, that has benefits for the individual, but also for the wider society as well, because relieves some of the burden on the healthcare systems that we see across the globe today, not just in the US and UK. One of the things that we've been quite intentional about, which also touches to some of the points that Heidi made, was we recognized very early on these big biobanks, these big data sets they generate so many discoveries. It's impossible for one company, whether it's industry, whether it's academic organization, to really maximize the potential of all those discoveries. So we were very intentional about publishing the discoveries, the summary statistics on a public portal that then allows the community across the globe to benefit from those and really focus more on the translational research component, which is, for me, the bigger bottleneck. And one of the things that are really rewarding for us is when we look at the access to these portals. Over 100 countries across the globe, we get over 10,000 users a month, and about 50 or so contacts from academics all around the globe, not just from US, UK, China, but in parts of the world that I never would have imagined people would come across the portal, reaching out and saying, we found something interesting, we've got a mouse model, we'd like to collaborate. It just catalyzes this kind of collaboration, global collaboration, to help us advance some of these discoveries towards translational medicines and therapies, rather than trying to do it all within one unit, whether it's industry or academic. And I think this has been really pivotal for not just industry, but again, there's a lot of benefits that can be demonstrated from sharing these insights globally across the communities.
It seems like both of you are hitting on this core theme of data sharing and openness. Are there things that we can be doing today to push that further? Because it sounds like it's come a long way, but what's holding us back from taking that even further?
Lawyers. No, joking aside, it is important that when you start a study that you really think hard about the consent and the data use allowances for that cohort that you're enrolling. And if, in the clinical lab arena where you don't have direct access to the patients and you're not actually consenting them to think about your requisition form and what are your, what you say you do with that data, and don't take the most cautious approach, because the patients actually want you to share their data and they want a diagnosis. And unfortunately, the decision makers are often the lawyers in your company or your institution, and not the patients who are like, what do you mean you're not actually sharing my data? So I think we all have a responsibility to think very broadly when we start our studies and we think about the data we're collecting, to get the patient's opinions in that game, and then abide by it and figure out how you're going to make that data broadly accessible. And it may require creative ways. Like right now, we're trying to share, rare disease data across every country in the world, and there are, national laws that prohibit data from crossing country boundaries. But we're setting up these federated query systems that allow us to get, access to a variant and a phenotype in a patient without actually sending a whole genome across a country's boundary. So, you sometimes have to be creative, but make the most of that data that you spend a lot of time collecting and on behalf of the patients who've given you that data.
I mean, I can only echo what Heidi said, especially around, you're thinking about these research participants. They're doing one of the most altruistic things they could possibly do, because they're providing their personal, data, their health records, their genetics data, to advance scientific knowledge, recognizing that in their lifetime they probably won't personally benefit from it. In a lot of these settings, biobank settings and so forth. But they're doing it because they want to help the future generations in their families, or future generations of patients who'll go through the same journeys that they've gone through. And to that point, I think we need to find a path forward to ensure that we're not the blockers of being able to use that data for advancing medical and human research. Of course, everything has to be anchored on the consent. That goes without saying. But when you go beyond that, then it's about how do we make it as accessible as possible into the hands of as many scientists as possible to be able to collectively as a community advance. One of the greatest examples of this, I think, is the UK Biobank in terms of their model for data access.
Yeah, the UK Biobank is a good segue into discussion about data types beyond genetics, because in addition to data sharing, they've done an amazing job on really driving the next wave of omics, proteomics. They've done amazing imaging studies. I want to ask about proteomics first, so maybe we'll start with you, Slavé. You've done some really interesting work looking at the plasma proteome and how that correlates years or decades in advance of disease. I'd love for you to just summarize that for the audience. Then Heidi, have your perspective on how that plays a role in in research and clinical care going forward as well.
Yeah, so happy to share. It is quite funny when I hear myself say it, but when we did the pilot of 50,000 plasma proteomes, right, a pilot. Of course, it was a pilot because now we're doing the full 500,000 plus an additional 100,000 who have repeat specimens collected as part of the imaging component of the UK Biobank. But that first 50,000, I would say, I was interested in the proteomics data, but lukewarm excited. I didn't know what to expect, to be honest, because it's plasma proteomics. And for a long time as a community, we're like, well, you need to access the exact tissue, the liver biopsy, the lung biopsy. And that's still very true for a lot of applications. But one of the things that was quite remarkable was that even within the plasma proteome, we can see very clear signatures of early disease in organ systems that are not blood-based. So Alzheimer's disease, things like prostate cancer, COPD, kidney disease, and so forth. So last year, I think it was last year, we published a tool called Milton, which is an AI model that we developed in AstraZeneca, which leverages the genomics, proteomics, and routine biomarker data from hundreds of thousands of individuals in the UK Biobank to develop AI models that can successfully predict the onset of about 1,000 disease endpoints. But remarkably, sometimes 15 years before they were clinically recognized. And this is, again, a real testament to the study design, because in the UK Biobank, the specimens were collected 15 years ago. And so we're profiling what the plasma proteome looked like in these individuals 15 years ago. And now we have the benefit of 15 years of health care follow-up, and we can identify signatures in those individuals from time zero that correspond with individuals who went on to develop COPD 8 years later, or blood cancer 4 years later. With fairly high accuracy. I think for 120 disease endpoints, it was 0.9 AUC, so 90% accuracy. So that has kind of really opened our eyes about the potential of plasma proteomics in being able to use as kind of a early disease, kind of pre-screening, screening application beyond maybe some of the obvious diseases you might think of when you think about what you can access from the blood. But again, I think the important thing there is that there's a lot happening in our body many years before we're symptomatic. And it seems like some of those signatures are detectable in the blood, sometimes a decade before it's clinically diagnosed, which is quite remarkable.
How do you think about that, Heidi? You probably have millions of samples stored where you may be able to go back and do this across the population you look after. How do you think about that as an opportunity? Yeah,
I think there's two different ways to think about this in the All of Us research program. So we are doing a lot of proteomic data generation as well as transcriptomic, other omic data. And I think that will be valuable in the same ways that Slavé just mentioned. But most of my time I actually spend in rare disease research, where we have much smaller numbers, and then each patient, is often a distinct disease. Most of the diseases that are still to be uncovered in rare disease are rarer than 1 in a million. So no single researcher, clinician, lab is going to have more than one case. So being able to do statistical analyses with omics data becomes a challenge in that regard. That said, I think some of the most promising technologies that we're seeing right now for aiding in rare disease diagnosis are transcriptomics, the RNA-seq application, as well as methylation analysis. We're starting to see signatures of rare diseases in the genomic, regions beyond the sort of classic Prader-Willi, Angelman, type of disorders that have always been around in that realm. And so I think and certainly the use of tools like Splice AI that are much improved at detecting splice disruption, including cryptic splice sites deep into introns, what we really didn't have access to before. The ability to then follow that with RNA-seq or correlate genomic data with RNA-seq data has allowed us to make a huge number of diagnoses by enabling that additional evidence from transcriptomic data. And it's sometimes challenging when you can't get a biopsy of disease-relevant tissue, but if you have the predictors like Space AI that can tell you where to look, then sometimes even, blood or fibroblast samples can, where you're looking, are enough to find a small signal and even a gene that's not highly expressed in that tissue. So I think those are some of the areas in rare disease that are really showing promise. I think we'll eventually get there with the sort of proteomics, metabolomics stuff, but it's still a little noisy for these incredibly rare disease cases.
Yeah, that makes sense. I'd love to have both of your perspectives on AI and machine learning. This is, if we look at the abstracts for ASHG this year, I'm confident we'll see more AI and ML than ever before, possibly by an order of magnitude. Where are you most excited about it, and where do you think it's overhyped or maybe not going to deliver on the promise?
You want to start?
I can have a first crack. I think part of it is related to what I was discussing before. We talk about AI as kind of the technology advances, and I think they're important, but critically we have to always remember that it's the data that catalyzes these discoveries, and access to the right data at the right scale, right diversity, right clinical phenotyping, longitudinal, multimodal, all those things I think are the most valuable component. That said, some of these new things like generative AI and large language models, you can understand conceptually why they would be advantageous in things like biology, where you're effectively trying to fill a gap in a sentence, right? Like you're looking at patterns from the data and saying, okay, well, this is most likely, how we complete that sentence or the word that's missing. So I can see the potential. That the one, I guess, caveat maybe is that nature humbles us every day in terms of our perception of how much we know about human biology, right? There's papers every day that teach us something new about a gene or new about a pathway or new about a clinical disease endpoint, etc. So to assume that we have all the data and all the knowledge already, that we already know how human biology works, we're not there. And so where AI is most successful is when you have structured, very predictable data. I think for some parts of human biology, we may be close, but for a lot of them, there's just so many gaps. I think we need to keep generating the right data, ideally in the right tissues, the right organs, etc., so that we can fully leverage the potential of these effectively pattern recognition tools that allow us to detect patterns in this multimodal data. That's just impossible for us as humans, for most of us as humans, to be able to detect visually.
Yeah. I would completely agree. I think in healthcare, the use of AI in image data extraction, just some amazing work that's being done to be able to improve the diagnosis of patients and detection of disease and things like that. I think, some of the areas that we're also trying, and in some cases succeeding, in other cases failing. So the use of large language models to try to capture all of the published data that's out there. Like in rare disease, we want to get every bit of evidence for a variant, right? And the challenges with paywalls in the published literature, it's kind of a complete mess of how we as a society or a scientific community put out our information in the publication system. There's so many flaws with it. That is a challenging area, is to get that data and trying to extract it. On the other hand, where we've had more success is once you have the literature in hand, we spend inordinate amount of hours, curators, extracting information from the papers, and that is proving much more useful to use AI techniques to get that data and structure it out of, which is a little bit silly in that the data was structured before you published the paper. So why do we have to go through this cycle? Which comes back to my original premise of share your data in structured, useful ways, standardized from the start, and we won't have to play this silly game. But nonetheless, there's historical literature that, we will have to go back to things, and I think making it much more efficient to pull information out is certainly very exciting. And then, as Slavé said, when you have the data and it's there, making insights of it, it's just a ripe area for AI that is really exciting.
Maybe I'll just do a build. I think one of the reflections I have, obviously observing people using AI for things like life sciences biology, is that I think, to fully harness the potential of AI, as you were alluding to, Heidi, we kind of have to let go of our preconceived beliefs of how human biology works. We have to be more kind of, out of the box thinking and say, that doesn't agree with how I think things work, but let's run an experiment to trial it, rather than, well, that's just wrong, because that's not how I was trained or what I learned during my PhD or postdoc. So there is I think a big cultural component and it's just, in embracing it, but that also, to get to that point, you need to build the trust. You need to have good examples that it is actually adding value, and so it's kind of a catch-22.
You have both done a lot of work and have been great advocates for health equity in genomics. I'd love to get your thoughts on where we are today, but more importantly, what we can do to make sure that we continue to push the envelope on this to make sure that the advances that we talk about here are accessible to everyone.
Mm-hmm. Yeah, I mean, in terms of, let's say, genetic diagnosis, for example, the ability for every patient to have the highest likelihood of obtaining a genetic diagnosis is definitely dependent on data sharing, both in the sense of, and we've started this project we call federated NOMAD, because NOMAD is still highly populated by data of European ancestry. But when I interpret a genome, the first step that we all do is to filter out common variants that aren't going to cause rare disease. But each population has different common variation. And so data from Africa is going to help filter my genome for a European ancestry patient and vice versa, right? So we all need to give that diverse data to help in sort of use of genome annotation and interpretation. So we're now, since a lot of this data can't leave countries, we're working to get all of these countries to process their genomic data in the same way that we do the NOMAD processing at the Broad, and then share their aggregate statistics so we can merge that with the primary NOMAD database. And that will create a much more globally diverse data set that will help everybody's genome be interpreted, including people of European ancestry. So that's a critical thing. That said, that's kind of helping with the workload by filtering out stuff that's not relevant when we think about rare disease. In terms of actually making a diagnosis and have variants that we understand and can call pathogenic, that's about our rare disease recruitment efforts and making sure that we're recruiting individuals from every population. And that's where, different strategies of how you reach every patient. And, in our Rare Genomes project, and this is work that Monica Wojcik led, really trying to figure out ways to meet patients where they are. So we now have, recruitment assistants who are speaking multiple language. They work nights and weekends so that they can consent somebody at whatever time it's convenient for them. We send mobile phlebotomy to people's houses. There's different ways that you can strategize to enable everybody to participate, even when they come from very different places and abilities and things like that. But you have to be thoughtful about that. So by getting a diverse population of patients, you will then get diverse data and evidence so that everybody's variants can have the same underlying evidence support to be able to diagnose. So like in rare disease, I sort of think about it in those two dimensions.
And it's not too different in the more kind of common complex diseases as well. If I think about the simplest way to phrase it, to truly understand human biology, we have to study all humans. We can't just focus on one community and extrapolate that to the whole global community. So as part of that view, which I think is shared by most geneticists in the world, actually, we've been quite intentional in identifying and establishing partnerships across the globe in understudied and often by extension underserved communities to really help enhance the visibility of genomics diversity. Again, not just from an important ethical point of view, but also from a scientific point of view. We're making discoveries today in cohorts of non-European genetic ancestry that just wouldn't have been possible if we continued, even if we had another 500,000 Europeans. The frequency of those variants that are driving those signals are just virtually absent in Europeans. And so you just completely miss them, and then you don't need 1 million, you just need a couple 100,000. You see a signal very clearly in individuals of Mexican ancestry or South Asian or African ancestry. And there are many good examples of these discoveries. So I think it's critical, and like Heidi mentioned, even though the discoveries are made in one community, generally the discovery itself or the biology extrapolates. It's just it was more common in that population due to adaptation or various, maybe genetic drift even, various factors. So we have to understand that to really take these advances forward, especially in the world of AI, right? If we train our models, our AI models, disproportionately on one global community, we know what we can expect in terms of disparities in the application of those models then to global communities. So more so than ever, if anything, with these new emerging technologies, we need to ensure we've got a good balance of global patient communities in our data sets.
I want to have both of your perspective on therapeutics. With improvements in diagnostics, it opens the door to genetically targeted RNA, gene editing, gene therapies, and also small molecules and biologics. There have been some incredible breakthroughs, but there are also challenges. Gene therapies have stumbled in the last couple of years with safety issues as the volume has started to go up, but they're treating more and more patients. I'd love to get your perspective on what you predict or see going forward and what's having the most impact in your pipelines and with your patients today.
So, I think if you look back on things that are successful today, they all had a hiccup somewhere along the line because the technology wasn't quite, we were still learning. So a lot of the modalities, I think, carry a lot of great potential, but a lot of the newer modalities, we're still identifying how best to take advantage of them. And for example, beyond understanding the safety liabilities, making sure targeted delivery is a challenge. Right? How do we get these new modalities to just the organ that they need to get to? Right? That's a challenge for the whole community. And I think that's going to be something that would, once somebody breaks that kind of barrier, then it'll open up many opportunities for across industry. So I guess what I'm trying to say is for a lot of the newer modalities, we are still learning. The whole community is working through it. And if the positive thing is all modalities, even the ones that are well established today, had that period where we were learning about how best to get access, how best to make it as safe, and how best to kind of ensure that it reaches the right part of the human biology where it needs to target.
Yeah, there's some great headlines from the 80s declaring biologics a huge medicine failure. Right?
And look at it today.
Right. What do you think?
Yeah, I mean, we're definitely in an exciting era. Like when the Human Genome Project was completed, it just, everybody thought that the next day, like there'd be genetic therapeutics. And that did not turn out to be the case. It's been a long journey since. But I do think we're in an era now where we are starting to really see, an upward trajectory in terms of genetic therapeutics. That said, as Slavé pointed out, there are definitely some challenges in terms of safety. And it's going to, in the rare disease space, I think there needs to be more collaboration and cooperation in terms of how do you tackle rare diseases where the number of patients that are going to benefit from any individual therapy are so small that most biotech, companies aren't going to want to invest. And a project we've been working on, Sam Baxter, who's a genetic counselor in our group, has been leading this, called Genie, a prevalence calculator. And it's literally working with rare disease organizations to help them figure out the prevalence of their disorder, because it's often the single most important thing to go to a biotech and say, actually, the disease that my kid has and our organization have, it's actually more common than you think. Here's the data. This is how common it is, and helping, convince the, company to actually pursue that with therapeutic development. So I think there's things we have to work on. I think the FDA has made some progress in terms of allowance for rare disease therapeutics and exceptions from some of the very steep requirements, but still there's safety issues there that really need to be thoughtful and dealt with, so that we don't sort of stall ourselves with mistakes that cost people's lives.
I'm going to ask one more question, and then we'll open it to the audience for questions. So if you have any in mind, then we'll just ask you to raise your hand in a minute. For people in the audience who are either early in their career or later in their career but thinking about a new chapter, I want to ask you all hypothetical. If you were to start over today and start a new chapter, what would you focus on? What's most exciting and promising to you that you'd, and that they did not prepare for this question, so I'm being very unfair here.
Heidi's looking at me, so I guess I'll go first. Give Heidi some time to have a better response. Well, I think clearly one of the biggest challenges we have at the moment, I mean, thinking from a kind of a drug discovery and patient-centric focus point, is not so much target discovery, I would argue. I think we have a plethora of great targets. I think it's more about going from a good target where you've got a clear line of sight to clinical outcome to understanding the actual mechanisms by which that target is acting on the human biology, because that understanding is then what kind of catalyzes the preclinical research, and it gives you an anchor towards something that you can develop medicines on. And this is where integrating kind of the causal genomics anchor with other omics modalities to better understand the human biology is really a hot area of interest. The other thing I would just comment on, because it's just a general comment, especially in the world we live in today with ChatGPT and so forth. Historically, that we had a lot of value from kind of like knowledge practitioners, people who synthesize a lot of literature, and they kind of can bring the papers together and discuss those discoveries. I think they play a critical role in any team and organization, but increasingly I think the value will come also from the talent in the teams that are more open-minded and curious and kind of question what we think we understand, because we're at the cusp of generating such large volumes of data. We don't need to make assumptions anymore. We can actually look at the data or generate the data if we get access to the right patients to really get empirical evidence, rather than this is how I think this gene is causing that disease, and let's run that experiment. Didn't work, okay, the gene's wrong. No, maybe our hypothesis wasn't quite right.
But was your question would we do something different if we were back at the first step in our careers?
Your CV is erased. You're starting fresh today. Where do you go?
I think I would make almost every decision. Well, it's interesting. I tell this story. I've never interviewed for a job in my life. Every next step has been some networking opportunity, hey, come be here, or come do this or do that. Despite that, I wouldn't have made any decisions differently, with one exception, and that is a better background in data sciences and computational, science, which I, never got. So I depend on an amazing team that works with me that has all those skills. I think I would have spent a little more time getting the data sciences background, given the era we live in. Otherwise, all of those random decisions, I stick by. It's like a wonderful world I feel like we live in.
That's good. No regrets.
No
All right.
regrets.
Do we have any questions in the audience? Be brave. Raise those hands.
Hi, thank you. Thank you very much. That was really interesting. So, Heidi, just on what you were saying there regarding the access to things like the computational training, do you think we need a space for people that are more later in their careers to learn those skills, and is that possible? Because I can definitely empathize with that. I did a degree in biology, and I'm being hampered a bit now by not having those computational skills quite to the same extent. Do you think that's possible to integrate at a mid or later career stage?
Before you go, I'm just going to repeat the question just in case anybody didn't hear it or we don't get it on recording. So the great question was, for people who are maybe midway through their career or and haven't had the chance to get the computational training, what advice or what resources might there be for people to do that in the middle of their journey today?
I definitely think it's very feasible for someone today to go back. In fact, Sam Baxter, who I mentioned, a genetic counselor in my team, actually went and took Python coding course and is, beefed up her skills in that area. For me, as much as I say that's my one regret, with the amount of time I have right now, it would have to be a deliberate choice of what do I give up to go, because you can't just go take a course and then and think you're just going to become proficient, right? It's not just taking the training, but then doing and using those skills. And at my current stage of my career where I spend, at least 40 hours a week on Zoom calls and in meetings, like there's no time to actually use the skills that I might learn. So for me, myself, I've come to grips with the fact that I'm going to be reliant on other people to do that stuff. But I think someone who's earlier in their career and might, or just wants to make a fundamental change in their career, absolutely, like it's never too late, pursue your passion. And, but you just got to dedicate to it and carve out time for it. I mean, I remember when next gen sequencing came out, I'm 54. It's been a while. And, I remember like, oh my God, like I'm never going to learn how this works. Right. Well, then we launched the technology in the lab and we offered sequencing tests, and I learned all the nitty gritty stuff around next generation sequencing, which seemed like so foreign when we're all used to Sanger in that era and this new technology. But, if you're going to use it, if you use it, you learn about it, right? So I think it's more than just, can I take a course and be trained? Am I going to shift my focus and use that? Because most of your knowledge is going to be generated in the doing part of it, not the learning part. So you have to make a decision that you're going to go that path.
So I might just add a bit, and it'll be interesting to see how the audience takes it. So I think a key part now, if someone in your situation, I would suggest a key part is actually thinking about what are the killer questions, like, kind of conceptualizing what are the questions that the data or the technology can help answer today that you couldn't before, and prioritize that, plus maybe do some training on prompt engineering. Because I will say, I've been actually quite impressed at the coding skills of these technologies. And so, you could get a pretty good prototype. And then, of course, you want someone who's a proficient expert to scale it and make it more robust and so forth. But, to generate a prototype tool, I think the key part is the ideation of, what is the killer question, what's the gap I'm trying to solve, and how can I use this data to solve it? And that's probably more valuable.
I would second that.
Hi, Sarah Senham from Mayo Clinic. My question is about collaboration and duplicative efforts. So has there been a method that has worked in your careers to identify collaborators who may be working on similar topics, and being able to really foster those collaborations and prevent duplicative efforts so we can actually get all this data quicker without feeling like we're competing with each other?
A great question. So how do you identify collaborators that you're going to result in a 1 + 1 = 3 situation and not duplicate efforts? Great question.
I mean, honestly, I spend most of my time like trying to deal with that very question. And, early on when I was, launched a brand new clinical lab and then realized just the amount of time my team was spending searching the literature on evidence about variants and, interpreting them. And so were all of the other clinical labs doing the exact same thing. And it was like, this is ridiculous, the amount of time we're spending. Let's just all share our classifications and the underlying evidence and the papers we found and all that data in a centralized place so that we can crowdsource the problem, right? And that's exactly what ClinVar became. There's still some, challenges there with people putting all of their evidence in their submissions, but none. And that's why I'm working now on this variant level matching, federated network where the actual patient data could be shared. But it's 100% for the reason that you cite. That is, there is so much duplicative effort out there. When we launched Matchmaker Exchange, different groups had the same candidates. But there's nothing they could do with them without sufficient evidence. So we match them together, and then they jointly work on getting those discoveries over the finish line. Almost every rare disease gene discovery we've made in the last decade has benefited from matches on Matchmaker Exchange that's brought us together with another group studying the same gene and having the same candidate. So, and then we do model matcher to match with functional studies that are studying the same. So any way that I can come up with to get people to work together on a problem instead of separate. I mean, there's some times when competition, energizes our field to push things forward. So I'm not saying like never, ever duplicate efforts. We want the sequencing technologies to compete sometimes, like get all those prices to come down, right? But at the same time, most of the time, I would say 90% of the time, we would do much better to work together on a problem than separately. So I 100% behind that.
And I can probably add from an industry perspective, I think if you look at some of the, and again, the UK Biobank is just one example, but one of the things that's really changed in the last decade or so is these pre-competitive consortia whereby like-minded institutes, for example, pharma partners who see the value in human genetics and see the value in these big cohorts, get together to fund the generation that couldn't be done by just one company. It's just not feasible. And so you get an organization, 14 partners, I think it is, for the proteomics data on the UK Biobank. And then that data benefits everybody. And we go our separate ways. Sometimes we partner on the analysis, but sometimes we go our separate ways. And I think that latter part is also important because I do believe that diversity in opinions on how best to analyze data is critical to science. If we all think the exact same way and we use the exact same tools all the time, it kind of stunts innovation. So we need that diversity of views of, well, I think we should use this tool, I think we should use that tool. And but the generation of the data is something that we all agree on. That's a valuable data set. Let's get together, let's generate the data, and then we'll go our separate ways in how we analyze.
We have time for one question, maybe two. Good. I think I saw a hand in the back there. Perfect.
Hi, my name is Vince from 5 Prime Sciences. I found the point on that Slavé made on mechanism of action and understanding that well was very pertinent. I would like to know the panel's opinion on how you see the future in understanding mechanism of action, maybe data sets, algorithms, or just your general insights on what would be useful to furthering that endeavor.
Great. Yeah. So what are the most useful technologies or approaches to better understand mechanism of action? Slavé, we'll start with you and then Heidi.
Yeah, I think from a technologies viewpoint, we've already got a lot of good enough technologies, and there will be better technologies that come across, Petrovski and various other technologies. I think for me, the biggest barrier that we often have is access to the right models, the right translatable models that we can use kind of preclinically. So, some kind of almost a concerted global catalog of all the models that everybody's generated that are available across the communities that one could then identify, okay, I'm going to go contact that collaborator in Tokyo because they've got a model that is relevant. That would be quite precious, I think, for a lot of the activities we do, rather than having to think about how do we build these models every time we have a new question.
Yeah, I mean, we're spending a lot more time in the rare disease space thinking about mechanism of pathogenicity because it's really helpful to interpret variation if you understand the mechanism of pathogenicity. Is it a gain of function, dominant, negative, loss of function? And then if you understand that, you can drive improved interpretation of variants that you find that fall into those categories. You don't always know in advance, but also in terms of validating and calibrating high throughput functional assays, MAVE assays, multiplex assays of variant effect. These, really need to understand the mechanism. So you take variants that are of the same class and use that to validate those algorithms because they're going to work differently if they're trying to predict a gain of function mutation versus a loss of function or protein folding impacts, things like that. So I do think even in the space of just variant interpretation mechanism, makes a difference in terms of understanding that, let alone thinking about therapeutic targets and really understanding disease biology so that you can intervene, which is a whole other area.
And one of the areas where it's been really successful is in cancers, because often in cancers, the cellular phenotype you're looking for is death or persistence. But for a lot of common chronic diseases, it's more complex and more nuanced than that. And having the right biological effect assay is kind of the biggest bottleneck at the moment, I'd say.
Yeah, super question. We have time for one more. I think he was first. Sorry.
Hey, great talk. With so much data being generated worldwide across many different institutions, how do you think about creating like normalization or standardization in order to use the data and compare it across different countries and institutions and so on?
Yeah, so I spend a fair bit of my time as the chair of the Global Alliance for Genomics and Health, and it is a standards building organization solely for the purpose that you just stated, which is there's no way we can benefit from data sharing around the world if everybody's data is in a different format, collected with different standards, all those things. So we have work streams across every dimension, whether it's the development of APIs to access that data, or the file formats for that data, or our genomic knowledge standards around the annotations of that data, nomenclature for calling variants. We have, quality for whole genome sequencing, like everything, and also our regulatory and ethics in terms of consent policies and data access policies, how we authenticate researchers to access the data. There's just 1,000 dimensions around how we're actually going to make use of this global, network of data. But there's so many things that we have to harmonize and standardize to make that actually happen. So if you're not already involved in the Global Alliance, come join us.
I've got nothing to add. Heidi's the expert in that as the chair of the Global Alliance.
I think that's a great note to end on. I want to say thank you all for the great questions and spending time listening to us, but a huge thanks to Slavé and Heidi for taking the time to be with us today. And if you liked this and want more of it, then you can find us on your favorite podcast player.
Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.